Thanks very much, and good afternoon. Bonjour à tous et toutes. I did my PhD in Orsay, so I have very happy memories of French science. I'm a physicist, but I'm actually going to talk about biology this evening, so that should be entertaining for all of us. And I'm going to talk about the physics of life. So when I was at school, I had a fabulous physics teacher. He was almost infallible, but there was one thing he got seriously wrong. He said you can combine physics with chemistry, you can combine chemistry with biology, but you cannot combine physics with biology. And this was his sort of career's pep talk. And he saw the world pretty much like this, that you have physics, which is all to do with lasers and microchips and electrons and waves and all that sort of thing. And then you've got biology, which is to do with squidgy stuff and stuff that's alive and molecules of life. And that really there's a fairly impermeable divide between them. The methods are different, the problems are different, they've just got nothing to do with each other. And I think that is one of the things that has changed in the last 30 years or so. Now, biophysics is one of the hottest areas of physics to be in. And physicists are discovering that a lot of the tools, both experimental tools but also conceptual tools, that they used to apply to atoms and electrons and things like that, can now be applied to really important biological questions, like what causes cancer and how does a cell work. So this is the era of biophysics. So in this talk, what I'm going to do is just give you a little flavor of some of the things that are hot in biophysics. One of the problems, as those of us working at the science-religion interface find, is that anybody who wants to span two disciplines always ends up feeling like a professional in one of them and an amateur in the other. And that's how I feel here. I'm a professional physicist with amateur interests in biology. So if there are any real biologists in the audience, please be gentle with me. Equally, if there are any real theologians in the audience, please be gentle with me. That is the nature of crossing disciplines. You've got to take that risk. I'm going to talk about two different things. The first half is mainly what we call instrumentation. So in other words, the sorts of machines and experimental techniques that we use in the biology lab in order to discover the science of life. And I want to show you two or three machines that are absolutely key to today's scientific work in life, in biology, that have come directly from physics. So the first is what's called confocal microscopy. I'm just conscious I'm banging a little bit. Is that annoying? Are you picking up is it okay? Great, I'll fasten my jacket. That might stop the cables being touched. So confocal microscopy is an absolute workhorse now, but there was a time when it was a new emerging technique. And it's a laser-based microscope that's very good at looking at living systems. This is what they look like if you buy them today. But actually, the original patent goes back to 1955 from Marvin Minsky. And those of you in the AI world may recognize that name. He is he's often referred to as the founding father of AI. And what's interesting is before he sort of found his groove of what his main contribution was going to be, he just had time to do this completely sidebar thing of confocal microscopy, which has actually revolutionized biology as well. And I guess some people are just really talented at whatever they do. And the point of a confocal microscope compared to a normal microscope, there are two big differences. One is that quite often you illuminate with one color, one wavelength of laser, and then you observe at a different color. So you're looking for color shifts. And that's really important. That would get you as far as what's called a fluorescence microscope. To make it a proper confocal microscope, you also need a pair of pinholes in the microscope. And those pinholes draw on a wonderful bit of physics of optics, and they give the microscope a very sensitive depth resolution. So most microscopes are very good at resolving in the plane. You can see things side by side with a gap between them. But they tend to telescope all of the depth together. Now that's fine if you're looking at a microchip where all the action is on the surface. But if we cross that divide into the biological world, living things are more intrinsically three-dimensional. And so the first step towards observing living systems is being able to take planar slices through them so you can build up the true three-dimensional topography. And that's exactly what a confocal microscope does so well because of those two apertures. So for example, these are some images taken by confocal microscopy of a pollen grain. So these are the things that make you sneeze if you've got hay fever. And what you end up with is Z slices. So as we go through each image number, we are a few microns higher, and you're seeing just that plane. And what's great is if you have all of these slices, you can then put it into a computer, and it can then reconstruct the full three-dimensional shape. And that's what comes out. And that little chap there is a full pollen grain. It's about 40 microns across. For comparison, underneath is what you get if you use a non-confocal microscope. So this is what biologists were able to see in terms of pollen grains until Minsky did his stuff. Now this is what we see today. And you can understand that's a huge step forward in being able to observe. And if you can observe, you can begin to build models to explain, to understand. And it powers the whole scientific process. The reason I mentioned about using different wavelengths is really important. So these are some cells. These are called HMCB cells. And what we do here is we illuminate with one particular wavelength, and then we look for either the blue or for the red. And we can use fluorescent markers to bind to different parts of the cell and to then show up in different colors. So now we've moved beyond just seeing the shape of something to being actually able to identify different functions within the cell. So for example, in this image, the blue, sometimes called the DAPI channel, shows you the nucleus of the cell, whereas the red shows you the cytoplasm, the stuff around the nucleus. So we've now taken, as well as being able to do three-dimensional imaging, we've now been able to go the next step and actually do functional imaging, see what parts of the cell are where, what do they do. This is another example where we do, this is called a live dead stain, where we do the same thing in a confocal microscope, but we set up the fluorophores so that if they glow green, that means the cell is alive. We look for some of the metabolism products. We know the cell is alive and healthy. And if it's red, we know the cell is dead. And so we can look at such fundamental functions. Are you alive? Are you not alive on the microscopic scale? So in this particular image, all of the green smears, these are cells, and then the little red dots are the dead cells. And this is really important in your experiments to be able to work out if you've done something to your culture, have you killed everything? If you've plated it out from where it was incubated onto where you want to do your experiment, maybe it was unhappy on the experiment and they've all shriveled up. You can see that very quickly with a live dead stain. And then sort of the extreme sport of this is where you then bind that fluorophore, the thing that actually lights up, that we look for with a confocal microscope, to an antibody. And the antibody will then bind to specific proteins on the cell. So we can now begin to say exactly what is the cell made up of? Does it have that particular molecule on its surface? And if so, where? And this so-called immunocytochemistry is really powerful because we're now deep into the workings of the cell. We can actually see what's going on at a molecular level. So for example, this was some work that my former postdoc, Selma Luelio-Papicho, was involved with, a French-Algerian woman. And we came across some old work that suggested that patients with early-stage kidney cancer would exhibit enhanced levels of certain proteins in their urine. And we were interested to know, could we make a sort of like a COVID lateral flow test style assay to diagnose or identify people with early-stage cancer tumors just through sprinkling a bit of pee on it. And so this was an example where we used those fluorophores, and these are the concentrations of a protein called NMP22, which is one of the proteins associated with cancer. And the higher the concentration, the brighter green these little test discs glow. And this is all because of that laser physics of the confocal microscope that it's possible. I love this monument. This is in the former Eastern German city of Jena, which is one of the big centers of optics. It's a real place that physicists love to go and get excited. Carl Zeiss, the lens company, grew out of there. And There's wonderful physics, optics research there. If you look in, this monument is in honor of a physicist called Ernst Abbe. And if you look very closely on there, you'll see this little equation. Let's just zoom in on that. D equals lambda over 2n sine alpha. Every physicist knows that equation by heart. Anybody who tries to do microscopy, who tries to look at things, knows that equation by heart. And the two things they know about it are they hate it, and it is literally and metaphorically carved in stone. You cannot beat the Abbe limit. What it says is, for a given set of optical conditions, a given wavelength of light, a given angle of the light, there is a smallest size that you can see with an optical microscope. And it drives biologists crazy because that size, when you put typical numbers into that, is about the size that all the interesting stuff happens in physics of life, inside cells. And yet physicists say this equation is immutable. It is like the speed of light or conservation of energy. You cannot beat the Abbe limit. It comes from really fundamental diffraction physics. And so that's why I say it is carved in stone. Let me introduce you to this gentleman, Stefan Hell, a German-Romanian physicist. He was not satisfied with this idea that the Abbe limit stopped you from looking at the physics of life. He spent his entire career saying there must be a way around it. There must be a workaround. All other physicists said he was wasting his time. He couldn't get grants. He struggled on with tiny little bits of funding here and there because we said it's a fundamental law of nature. You cannot beat the Abbe limit. This is him receiving the Nobel Prize in 2014 for having beaten the Abbe limit. A remarkable piece of work. Let me just show you how good it is. This is called the nuclear envelope. This is the nucleus in the middle of a cell. This is the perimeter of that nucleus. And we've used our confocal microscope to look at different bits. So the red shows you nuclear pores, the green shows you the envelope, and the blue shows you chromatin. And it's a bit blurry because that's a micron, and if you put the numbers into the Abbe limit, it comes out at about half a micron. So we are right on the edge of what we can resolve. Let me show you exactly the same bit of cell looked at with Stefan's new microscope. It's amazing, isn't it? You can see these little nuclear pores. Instead of just being a tiny red smear here, you can actually see them precisely. This nuclear envelope, you can see it as a sharp edge as opposed to a diffuse blur. And this has just transformed biology. It's called super resolution, and it uses some really clever physics to beat the Abbe limit. And it allows you now to use light to look at things that are smaller than the wavelength of light and on a length scale that's relevant to the way cells work, to the way a lot of biology, a lot of life operates. What's next? Well, those of you who know the physics world will know everything is about quantum technology these days. It's one of the big, alongside artificial intelligence and advanced materials, this is the triad of hot topics. There are some really interesting ideas in quantum technology called quantum imaging. We already know that one of the foundational concepts in quantum mechanics is superposition and entanglement. The idea you can take two quantum objects, maybe something that has like in this figure, something that has two possible states, an up or a down, a 1 or a 0. You can bring them together, you can make them interact, you can do what's called entanglement, you can then separate them, and then if you observe this one, something happens to that one, even though they are not connected by any known mechanism. The fact they were quantum entangled at first means that even when separated, they have this strange link between them. It's been well exploited in cryptography, so we can use it, for example, to see if anybody has eavesdropped on secure conversations, secure communications. Some biophysicists have the idea we could use similar things to do new types of biological imaging, to be able to see into places that we currently can't see. This is the sorts of experiments you end up doing. Again, lots of lasers, lots of mirrors, lots of really physicsy things. But then you apply them to biological systems. So this, for example, is a section through a mouse heart. So the top one is the image obtained through a normal confocal microscope. This is obtained through quantum imaging. It's very early days. We don't quite know what the benefits are. I think at this stage all we know is we can do imaging using these strangely entangled photons. But potentially there's a lot of new things we could do. Let me show you a couple of examples of things that colleagues in Glasgow are working on. So in this one, for example, they're trying to use this connection between photons to look through the skull. Now, in principle, you can't use optical microscopy to do brain studies because the skull is too thick. The light doesn't get through. They have ideas that say systems like this could actually get inside the skull, and we could do optical imaging through thick bone. This one is a variant on that, and it says, well, think about an endoscope. An endoscope is a bundle of fiber optics that's about 1 millimeter in size, and actually on a medical scale, it's quite chunky. There are places you can't get an endoscope that you'd quite like to. Physicists already know how to make optical fibers that are 100 times smaller than that bundle. What we don't know is how to do imaging through just a single fiber. And so there are ideas you could use, again, quantum entangled properties to do imaging through a single fiber, which would then allow you to get into all parts of the body and have some really remarkable new imaging tools. So we've started with the confocal microscope. We've then progressed into the quantum realm. The ultimate for imaging, though, is electrons. And physicists have been using the transmission electron microscope and the scanning electron microscope for decades. The trouble is, it's really hard to use in biology because under that focused beam of electrons is a really high energy environment that's not conducive to life. You tend to just fry most biological systems if you try to put them in at least living systems, if you put them in an electron microscope. Another colleague of mine, Richard Henderson, fellow of Darwin College, my college, this is him receiving the Nobel Prize. And just like Stefan Hell before him refused to accept the limitation of Abbe's law, Richard refused to accept the limitation of electrons for use in living systems. And he, over a whole lifetime, developed a technique called cryo-TEM, where you essentially freeze biological material in a microscopic layer of ice that protects it from the high intensity of the electron beam and allows you to then put your biological samples into these monster electron microscopes and get atomic resolution. And structural biologists just love this. So if you remember during the pandemic, we all saw pictures of this coronavirus with these sharp spikes, the crowns. I don't know if you ever wondered, how do we know? How do we know what those viruses look like? Because they are about 100 nanometers in size. They're much smaller than the diffraction limit. Well, we know about it because of cryo-TEM. You can reconstruct biological entities like viruses. This is a phage, bacteriophage, with atomic precision as a result of Richard Henderson's work. Okay, that's all I'm going to say about instrumentation. Let me just take a few minutes to then flip to the other side and say, in what way are the conceptual tools of physics being used in problems to do with life? And a big area at the moment is mechanobiology. And that's really saying to what degree are forces important in living systems? Now, we all know if I bang my hand against that, the bruised knuckles is a result of forces. So yes, clearly we are sensitive to forces, but to what degree at a cellular level do we use forces as part of the way life operates? And I was involved with a project to say, well, are bacteria sensitive to vibration? Does the rate at which bacteria reproduce, is that affected by their mechanical environment? Because if it is, that gives us a whole new angle on antibiotics, for example. At the moment, we attack bacteria through chemical means, through bacteria. But what if we could use a slightly more physicsy thing of forces and vibrations? So we developed this little system, this vibrating cantilever. It's only 50 nanometers thick, and we can make it vibrate at a particular frequency. But otherwise, it looks just like a planar surface. And we cultured E. coli onto the surface. E. coli, they're the bugs that give you food poisoning, but they're a real staple of every biologist's lab. And they're pretty aggressive. You incubate a few E. coli on your experiment, and before long, they've just run riot. They've taken over everything. So what started as my nice, clean experiment, give it a few hours, and it's covered with all these E. coli that have just replicated time and time again. And they've actually set about my carefully built micro cantilevers. You know, they've started to twist them and to pull them apart. They're really aggressive. This was the control experiment. This was where we didn't vibrate them. Let me show you the actual test where we allow them to incubate in exactly the same conditions, but now we are vibrating those cantilevers with just a few microns of displacement. This is what you get. Unlike the static version, which has just been overrun with the bacteria, here we don't have that many more than what we started with. So for reasons we don't quite understand, their ability to divide is largely suppressed by these micron-scale vibrations. So they're clearly very sensitive to forces. There's a whole lot more that can be said. We've been looking at this as a potential cancer therapy. There are people who go into much greater depths of the understanding of what's going on within the cell, but it seems that very physicsy thing of force is key to how the cell works. If I had more time, I could show you a similar story to do with voltage. It seems that across a cell membrane, voltage is really important, and you don't get any more physicsy than voltage. That is a final thing. I just want to give you a little snapshot of my own research area. So I put most of my time at the moment into seeing what happens if you combine cells with microchips. So we use semiconductor industry techniques, the sort of things that people like Intel make microchips on, and we then coat them with cells. And then we activate a release mechanism that means the little microscopic chips float off from the wafer and form a suspension. So we've sort of made an artificial liquid where we got to design each of the little particles in that liquid using microchip methods. I call these little flying carpets for cells. Each little flying carpet can carry about 40 cells. But they're now in liquid. And once they're in liquid, we can do all the sorts of biological assays that biologists traditionally do. Let me just show you one of these liquids. One of the fun things we do is we incorporate a little bit of magnetism into the little microchips so that now, in liquid, they respond to a magnetic field. So the green are the cells. They're fluorescing, just so we can see them more clearly. And the darker squares, those are little microchips. And as we wave a bar magnet around this solution, you see all of the little flying carpets align with the magnetic field and move the cells around. So what we've now got is remote actuation of little clusters of cells. And this gives us a lot of potential. We can use it to discover drugs more quickly, more effectively. We can use it to diagnose cancer. We've got lots of different applications that are all possible once you have these cells in this mobile form and can remotely actuate them. I think I've got a minute left. Is that about? You happy with that? I've got 5. Excellent. I'll talk slowly for the last slide then. This is a session on science and God. Let me just give you a very brief theological reflection. I'm a Christian, and for me, science is a pious activity. It is part of my faith to engage with the scientific process. 2 things come to mind. One is from the Proverbs. It is the glory of God to conceal a matter. It is the glory, to search out a matter is the glory of kings. And I think that's really what I've been describing here. Physicists are providing the tools to help us in that glorious activity of seeking out what is the true nature of the creation, to be able to see more clearly the works of the Lord. That's how I see instrumentation fitting into the big picture. And also from the Psalms, I praise you because I'm fearfully and wonderfully made. Your works are wonderful. I know that full well. And I think that is the right response to the things that we see and that we observe once we've used all of these advanced tools to actually see that creation in more detail. I haven't had time to talk to you today about ATP synthase, the little micro robot that operates in every one of your cells, producing power to drive life itself. It's a remarkable thing. We are indeed fearfully and wonderfully made. With that, I'm going to say thank you for listening. Merci beaucoup.